Hello everyone, this is Richard from Modern Healthspan. Today we'll look at a possible NAD booster, which has not been talked about very much in the past. This is Trigonelline. I will use this study as a base. In the study, the authors showed that Trigonelline improved muscle function in aging and is lower in sarcopenia. The lead author on this paper is Dr. Vincenzo Sorrentino, now at the National University of Singapore, who we interviewed in January this year, and we spoke about this topic. I have linked to the interview above. Both mitochondrial dysfunction and low NAD levels are correlated with sarcopenia, but it's not clear whether these can be impacted by systemic changes or diet. In this study, the authors found that a natural alkaloid, trigonelline, is correlated with muscle strength and NAD levels in multiple species. Levels of trigonelline in the blood were reduced in people with sarcopenia and positively correlated with muscle strength and mitochondrial oxidative phosphorylation in muscle. There is no known path for trigonelline to convert to NAD. However, using labeled molecules, the authors showed that dietary trigonelline did end up in NAD in C. elegans mice and in human muscle cell cultures. Although the exact mechanism is not known, it was dependent on the Price handler NAD pathway. Although similar to nicotinic acid in chemical structure, trigonelline does not activate the same receptor which causes flushing with Na. They looked at a couple of in vivo models with C. elegans and mice of supplementation with trigonelline. In mice, it helped with muscle strength and reduced fatigue in aging, with the overall conclusion that supplementation with trigonelline boosts NAD and may help with age-associated muscle decline. So what is trigonelline? Here is the chemical structure. You may recognize this as it's very similar to nicotinic acid. With the addition of this methyl group, the molecule is a zwitterion, which means that it has both acidic and basic elements that react with each other, hence the negatively charged oxygen and the positively charged nitrogen. It is present in some plants, particularly in coffee and fenugreek, and gets its name from the scientific name of the latter, Trigonella phoenum graecum. The major indication that they were studying was sarcopenia, which is the functional loss of skeletal muscle with aging and leads to loss of mobility and physical independence. Clinically, it's characterized by loss of muscle mass and strength, leading to loss of function, such as gait speed. At the cellular level, this involves wasting of the muscle fibers and mitochondrial dysfunction. The first thing that they looked at was, are trigonelline levels in blood correlated with sarcopenia and its clinical characteristics in humans? The data for this graph came from a cohort study called Mimosa, which I think had 119 participants from various locations and ethnic groups. Serum trigonelline was significantly lower in the group diagnosed with sarcopenia. Here are the graphs for specific markers: limb muscle mass, grip strength, and gait speed. There is a similar correspondence for each one of these. And finally, higher trigonelline was associated with a higher expression of the proteins involved in oxidative phosphorylation, a key part of the process that mitochondria use in generating energy. One of the questions they looked at was whether the trigonelline was being directly incorporated into NAD. To do this, they used labeled trigonelline with mice. The trigonelline had a carbon-13 in the carbonyl group and 3 deuteriums placed in the methyl group. If the trigonelline was included in an NAD, we would expect the NAD would be N+1, as the methyl group would have been removed. They saw that NAD levels were raised in the left-hand graph, and then there was an increase in NAD at N+1 in the right-hand graph, showing that the pyridine ring is indeed directly included in NAD. They looked at liver, muscle, and blood NAD levels and saw a significant increase in each case. The authors performed a number of in vitro experiments to determine how trigonelline was incorporated into NAD, which I won't go into in detail. They did not come up with a complete answer, but it did seem to be through the Price handler pathway, as used by nicotinic acid, rather than the salvage pathway. As can be seen in earlier graph, NAD increased more than just the amount added by trigonelline, so it seems to promote NAD generation as well. They tried trigonelline in C. elegans, where it increased lifespan and healthspan, which I won't go into. They also performed an in vivo test. The improvement in muscles was mostly suspected to be due to the increased mitochondrial health, which normally deteriorates with age. They saw that the number of mitochondria did not increase, nor was there any change in muscle mass or other measures of body composition. However, there was an increase in the enzymes used in oxidative phosphorylation, the process that mitochondria use to make ATP. There are 4 protein complexes used in OXPHOS. These graphs show the activity of the first and second of these proteins in this process, showing an increase in activity. At the physical level, there was an increase in grip strength of around 15%. Older muscles fatigue more easily. In a test where the muscles were electrically stimulated, 50% of the fatigue loss with age was recovered with trigonelline. Here are the details of the mouse study. Young mice were 12 weeks old, equivalent to about a 23-year-old human, and the old mice were 20 months, around 60 years for a human. The trial lasted 12 weeks, and the mice were given 300 milligrams per kilogram per day trigonelline in their chow. Using allometric conversion, this would be about 24.4 milligrams per kilogram for a human, or 1.8 grams for a 75-kilogram person. As always, these interspecies conversions are inexact, and just to give an idea of the magnitude. Where can trigonelline be found in natural sources? The highest concentration seems to be in coffee beans, at 6,000 to 13,000 parts per million, depending on the style. It is also present in fenugreek, which is available as a supplement from various suppliers. What kind of trigonelline dose would you get from these sources? As with the previous calculations, this is not meant to be exact, but just to get an idea of the order of magnitude. I was not able to calculate what parts per million would mean in terms of drinking coffee, but I did find some sources which may be able to help. This paper, despite its title, looked at trigonelline levels in coffee beans. Green Arabica coffee beans have between 9.87 and 12.6 milligrams per gram, whereas a cup of Arabica espresso has around 70 milligrams per cup. Roasting conditions affect the amount of trigonelline in the coffee. Though it is interesting that decaffeinating coffee does not significantly reduce the trigonelline level. There was also one human trial which directly tested the effects of trigonelline on humans, in this case studying the impact on glucose tolerance and insulin levels. A couple of things to note here. The dose for the intervention with trigonelline was 500 milligrams, and a cup of coffee provided 72 milligrams of trigonelline, which agrees with the previous paper. In this study, the trigonelline and the chlorogenic acid arms produced a significant result, but the coffee did not. Unfortunately, they did not look at the level of trigonelline in the blood in each of these arms. However, it seems that it would be difficult to reach the same level of trigonelline as the 500 milligram intervention through drinking coffee. However, coffee does raise trigonelline levels in the blood, which can be used as a marker to estimate the coffee consumption, because trigonelline is quite stable in blood, unlike NR and NMN, which are quickly degraded to nicotinamide. One final point is that drinking coffee has been associated with lower levels of sarcopenia, so maybe the trigonelline is having an effect. How about fenugreek? I found this paper which looked at trigonelline yields from fenugreek seeds. Here are the key results. This graph is showing grams of trigonelline per kilo of fenugreek seeds. The study was looking at growing conditions such as irrigation and fertilizer, which is why we see different yields, but my rough estimate is about 3.1 grams per kilo on average. Searching on the internet, a common size for a fenugreek capsule is 500 milligrams, so this would contain about 1.55 milligrams of trigonelline. So this looks like it would be difficult to be able to reach the tested levels of trigonelline in the previous study. There are other possible sources of trigonelline. It's possible that it is made by the gut microbiome, and it is also associated with folate and fiber intake. One last thing. As we discussed at the beginning, trigonelline is very similar to nicotinic acid. NA has the effect of causing flushing when taken in high doses. Does trigonelline have the same effect? The flushing is caused by NA when it binds to a G protein-coupled receptor 109A, which causes the blood vessels to relax in the skin. The authors tested this in vitro and saw that trigonelline does not activate this receptor, so should not have the same effect. In conclusions, the authors point out that trigonelline helps with fatigue and grip strength, but did not help with muscle mass and should be combined with nutrients which support that, such as protein, vitamin D, and omega-3 fats. Also, at the cellular level, the improvement was not due to structural changes in the muscle, but rather to the improvement in mitochondrial respiration. It was also shown to raise NAD and have similar benefits to NR and NMN in preclinical models. It is therefore a possible NAD booster. However, research is very early and there is little data on dosage. But it does seem like a good reason to keep on drinking coffee. Thank you for your attention, and I wish you all well.